is Professor Niccolo ZAMPIERI. No, he's not professor, it's just Niccolo ZAMPIERI. He is from, and here my German would be not good, Max-Delbrück, now without the T, Max-Delbrück-Centrum, Aoso, Solala, Sonzo, Berlin, and he will explain us, tell us something like sensing from within, an intraspinal sensory system for motor control. The stage is yours.
Thank you. Can you hear me? Can you hear me? Yes. Okay. Thank you, Gaspare. Thank you for having me. Sorry for changing the title. Obviously, I'm a little bit an outsider here, and I'm very excited to be around human physiologist. I'm a developmental neurobiologist by training, and I'm interested in understanding development and function of neural circuits that control movement. And all my work is done using mouse, but obviously for me it's very interesting to learn from human physiology. So as you can see from my beautiful dog, animals can execute also a variety of different motor behaviors in a seamless and almost effortless fashion. However, we still do not quite understand the neural mechanism underlying robust, adaptive, and flexible motor control. This is a very complex problem, but if you wish, at its core, it boils down to one simple task that the nervous system has to do, which is a transformation of sensation into action. And at least if you think about voluntary motor control, there are two essential ingredients for this transformation that are for the nervous system to generate a representation of a sensory representation of the internal state of the body and the surrounding environment, and generate a motor plan in order to execute the desired action. And again, in a very reductionist way, you can think that the sensory input has a very central role, because sensory information is crucial to inform the generation of the motor plan at supraspinal level in the brain, but is also used online to modulate ongoing motor command, and this mostly happens at the level of the spinal cord, where the executory circuits are located. In my laboratory, we are really interested in understanding what happens in the spinal cord and how spinal sensory motor circuits orchestrate movement by integrating motor commands and sensory information. And we particularly focus on somatosensory input because it has a privileged access to the spinal cord, and most of sensory input gets to motor neurons in mono or disynaptic pathways. And obviously it is known from patients that suffer from severe peripheral neuropathies that movement is almost impossible without somatosensation. And patients like this one, they lose large diameter sensory afferents that carry information from mechanoreceptive organs that are located in the skin and relay different flavors of tactile information, and in the muscles, where they relay information from muscle spindles, information about stretch, logic tendonal organs that detects force. And this is critical because the nervous system somehow uses this information to estimate the body position and movement in space, which obviously is super important for controlling movement. We take advantage of mouse genetics to find a way to perturb selectively different sources of sensory information, and then assess the result at the level of motor behaviors, to establish causality between circuits carrying different sensory information and their role in motor control. We use as a quantitative way to study movement treadmill locomotion in a mouse, which is basically everything that happens in treadmill locomotion in a human, miniaturized. We extract hundreds of motor parameters that then can be used to assess to compare treadmill trials and different conditions. And we couple this with the power of mouse genetics, which I won't go into, but we have very interesting tricks that allow us to insert into sensory neurons of choice different molecules that allow us to visualize the neurons or perturb their function, or such in this case, we put in the diphtheria toxin receptor. This is a receptor that is present in human mice for the diphtheria toxin at very high affinity. Diphtheria toxin kills whatever expresses the diphtheria toxin receptor. In these animal sensory neurons that are connected to muscle proprioceptors, muscle spindle and Golgi tendon organs, express the diphtheria toxin receptor. If the animal lives happily all his life until you give diphtheria toxin. When you give diphtheria toxin, you have rapid and efficient ablation of proprioceptive neurons, as you can see here. DT0, we give diphtheria toxin 1 day after injection, about 25% of neurons are lost, and so on and so forth. And at the same time, we can study locomotion of the animal on the treadmill at different time points. And as you can see, already 3 days after diphtheria toxin administration, the animal locomotion is widely impaired. Then we can use quantitative analysis of motor parameters to sort of define the changes that happens in motor parameters between before and after diphtheria toxin administration, which sort of these changes, you can see them as motor signatures of proprioceptive sensory neural loss. And I won't go into the details, I just want to give you two take-home messages. The first one is the following: if I look at how these changes happen over time, the changes in motor parameters nicely scales with the amount of sensory neural loss, then confirming that there is causal correlation between loss of sensory neurons and phenotypic behavior. And the second one, if you look at animal before DT3, when the apparent locomotor behavior deficits are evident, the animal seems normal. And so the changes in signature precede the changes in the locomotor phenotype. For us, this is really exciting. And so in the last minute, I just want to give you a short summary. We have identified motor signature of proprioceptor loss, which we think are very important for studying the role of proprioception in motor control. The system is robust and can produce effective locomotion even after loss of 50% of proprioceptor. This is at 48 hours after diphtheria toxin injection. And finally, the changes in motor signatures precede the locomotor phenotype, which means they have a predictive value and can be seen as biomarkers. And now here I'm going to little bit stretch for this audience, but if you think that the neural mechanisms controlling movement, they are fundamentally concerned between mouse and humans. And so maybe we can learn something from these motor signatures that can be applied to human physiology. What are we doing with this? Sorry. We are asking, is there functional recovery from loss of muscle proprioception? And so we have been following now these animals for up to 60 days. And yes, there is functional recovery, but only if you train the animals. Very interesting data from Alessandro. Is robustness due to compensation from other sensory input? We are looking at the role specific of tactile feedback. Can we originate, identify signature of different physiological states? And for instance, we are very interested in aging, and since this morning many people talked about aging, we are having a very huge study in mouse, just started, where we follow the mouse longitudinally throughout his life, do a lot of behavioral characterization and molecular characterization. And can we take advantage of the mouse model as a flexible model to use in mouse genetics, different lifestyle, different social conditions? If you are interested and you have questions and you want to help us, please reach out.
Yes, a long flight. Questions? More than one, so ask at the end. Mine is not a question but a remark. I was referring in my presentation to the work of Gandilia, where he eliminated the apparent input by anesthetizing the sensory nerve.
And he found that it was not the maximal contraction that was, it was the maximal contraction that was impaired, but the submaximal contraction was not impaired. This is quite interesting, because in the case of your patients, in most daily life, they use some maximal contractions. And of course, what matters most is not only maximum force, but the precision of movement and the ability to modulate force finally. And this is the biggest impact on motor function, I think.
Yeah, I totally agree. I mean, I think, well, first of all, here we eliminate everything, and there are other tricks that will allow us in the future to go and ask more precise question, what is the role of 1B, 2 and 1As, which is really interesting. It is speculated, it's not really known. And when you can have this precision, then you can really dissect what is the contribution, and also you can test in different kind of movements, because obviously different movements have different requirements. And so this is really just the start that can allow you to really dig in and understand how different sensory information informs the system and allows the system to do what it needs to do. And I also think the role of tactile information is really important. We have evidence that as soon as you remove a little bit of tactile information, there is sudden degradation, even much more than what we see, which suggests that there can be a lot of compensation, which can be exciting for rehabilitation strategies and all of that.
Of course, thank you. Last very quick question upstairs.
You have to climb.
Okay, thank you. Hopefully quick. So I have another study in mind by Marcus Amann, by the group of Jared Dempsey. They studied cyclists with lidocaine injection L4, L5, cutting out the afferents but without affecting the motor control. They found that they were perfectly able to cycle, to do the same performance, but at a higher oxygen consumption and higher EMG or mean square. So this could suggest also a different motor unit recruitment pattern without or with less sensory feedback.
Yeah, absolutely. I mean, as it was alluded before, the role of sensory input is huge in motor neuron recruitment and in which synergies are recruited and in which temporal order. And I think that is very relevant. And I think maybe Alessandro later on will have something to say about this. Thank you.
Thank you very much.